The topic selected is virus, the pathogenic microorganism. The definition is it is a parasite that is incapable of reproduction alone but are highly infectious. The sentence has two parts. Incapable of reproduction alone means if you keep it outside, it will never reproduce. That is, for example, you keep it in nutrient agar medium, it will never reproduce. The second part is it is highly infectious. A microorganism or an organism can be infectious only if it multiplies. That means after going into the host, the virus multiplies or reproduces and then causes infection. Now, the virus, what does this mean? The virus means poison. The structure of the virus has two main parts. One is the nucleic acid and the other is a protein coat that is called capsid. The nucleic acid is RNA or DNA and it can be single stranded or double stranded. Now I have given the structure of two different types of viruses. One is the naked virus. This is the naked virus. Why it is called naked? Because only the nucleic material and the protein coat is present. That is the capsid. And together we call it as nucleocapsid. The other one is the enveloped virus. Why it is enveloped virus? Because the nucleocapsid has an outer coating of phospholipid which has the glycoprotein spikes. So, here we see that the virus has a virion. What is a virion then? The virion means it is that part of the virus that is capable of causing infection. That is nucleic acid and the capsid that causes the infection. Okay. Now, according to David Baltimore, the virus has been classified into 6 different types. Coming back to the capsid we said is made up of protein. But how many proteins are there? It has been found either one protein or it can be very few proteins. But multiple copies of the proteins are there and each protein encodes a single gene. And it has been found that those limited genes are capable of forming a large capsid. Now, as we stated that virus cause infection or it is pathogenic, that means it requires a host where it will go and multiply. Now, the host is of very narrow range. One is bacteria, that is the bacteriophage. The plants, where the plant viruses attack. We will concentrate on the animal viruses because in the recent times the topic of discussion is virus, and the recent viruses that are seen nowadays are SARS coronavirus, the COVID-19 virus, Ebola virus, and apart from that one of the very old one is the influenza A virus. Now, one characteristics of the animal virus has been found that normally animal virus they affect the similar species. That means, suppose a virus is infecting a human, for example, influenza A virus. Normally, it is restricted to human only. It does not go to the birds or the reptiles, unless and until the mutation has taken place. Why this is happening? Because virion, the infectious material of the virus, it has specific receptors where it can bind into the host cell. As it is highly specific, so it can attack the close related species. As we were talking about the animal viruses, here we come to the structure of various animal viruses. And if you see the coronavirus, the hepatitis virus, the Ebola virus, HIV, influenza A, if we see that all of these viruses are enveloped, all animal viruses are enveloped with the glycoprotein spikes on them. And this glycoprotein spikes, what they do, helps in the attachment of the virus to the host cell. So, how do the virus attack a host cell or grow into the host cell? It is very simple process of attacking. Now, how it occurs? Now, first thing happens is the virus, they attaches to the receptor, enters into the cytoplasm, and what happens? They form the multiple copies of the proteins are there, so they can attack the receptors. After entering into the cell cytoplasm, they what happens? The virus or the viral nucleic acid moves out and the phospholipid layer is removed. Now this viral nucleic acid now go and enters into the DNA of the animal nucleus, and what they do is they start synthesizing the mRNA into the host cell using the host cell enzymes, using the host mechanism, and then the viral mRNA is formed. And this mRNA that is formed, now they form the new progeny or the new generation of the virus. But how is this replication possible, or why the host body allow the replication of the virus? The three things plays the major role. First is there are certain enzymes that are present in the virus that helps in this process of replication. Second is there are certain inhibitory factors inside the virus that will stop the DNA, the RNA and protein synthesis of the host cell. So I am repeating once more that in first case what happens, the virus enters, they have the enzyme and they help in replication in the host cell. The second thing is that the host cell has DNA, RNA and protein synthesis process. The virus has certain inhibitory factors that stop this process. The third thing is that there are certain structural proteins inside the virus. After entering into the host cell, they help in the virion synthesis. Now two things happens. If it is a naked virus, normally the virion that is formed, that is the nucleic acid and the capsid, they assemble and they come out of the plasma membrane of the host cell by bursting the plasma membrane. That leads to the rupture of the host cell. But if it is an enveloped virus like in the animal cell, that is affecting the animal cell, then normally budding occurs. That is part of the plasma membrane forms the coat or the envelope and no rupture of the cell takes place. The growth cycle of the virus is called the lytic cycle or the lysogenic cycle. So here I will explain you the lysogenic cycle of an animal virus, that is rabies. Now we are seeing that there is the nucleic acid, that is the red one, there is a green capsid and the coating, the envelope with the blue glycoproteins. What happens? The virus comes near to the host cell. This is the host cell. It comes near to the host cell. The host cell has multiple receptors. The glycoprotein, they are specific to the receptor. They come and bind to these receptors. Due to endocytosis, they enter into the cell cytoplasm. The endosome organelle, they form a vesicle inside with the virus. Endosomes have certain characteristics. They take up the proton from the cytoplasm and inside them. When the proton enters the endosomes, what happens? The pH reduces. In this reduction of the pH, what happens? The glycoprotein layer of the virus breaks and the red capsid moves out. The red capsid or the red nucleic acid comes out. What the nucleic acid do? There is the presence of the viral RNA polymerase. The viral RNA polymerase now takes up the ribonucleoside triphosphate that is present in the cytoplasm of the host cell, and they replicate to form the viral RNA genome, and which further forms the mRNA. The mRNA that is formed by the viral mRNA that is formed have multiple functions. So what they do is, some of them encode for the glycoprotein synthesis. The mRNA that is responsible for glycoprotein synthesis moves to the endoplasmic reticulum. After going to the endoplasmic reticulum, they take up carbohydrate from the endoplasmic reticulum, become a mature glycoprotein, moves out of the endoplasmic reticulum and moves to the Golgi apparatus. In the Golgi apparatus, they attaches with the vesicles. After they attach with the vesicle, they come to the cell membrane, the plasma membrane of the host cell. What they do is, the glycoprotein is released on the plasma membrane of the host cell. Here you can see the blue color receptors. These are the glycoproteins of the virus. Now the other mRNA, what they do? They further go into replication and they form the matrix protein or the nucleocapsid protein and the viral RNA polymerase. Now all these things that are formed, they assemble together and they form a nucleocapsid, that is the new nucleocapsid, which is called the progeny capsid. This assembled progeny capsid now goes towards the cell membrane. What they do is, they take a part of the cell membrane that is having the glycoprotein and moves out of the cell. The cell remains intact, they do not break and do not burst, and this forms the new virus. Again that new virus goes to the next cell and they form the another progeny of the virus is formed and attack the host cell. In this process, the entire reproduction of the virus occurred and the virus remained inside the body. So this was about the how virus is entering into the host cell and the preliminary structure of the virus. These are the references from where the virus structure has been taken. Thank you.